Hello everyone, welcome back to part 2 of enzymes, and hopefully in this video we're going to be wrapping up this video, this entire chapter of enzymes. So before watching this video, make sure that you have watched the part 1 of enzyme, where we explained the entire mechanism of enzyme and what exactly are enzymes. Okay, so let's get started without wasting any time. And yeah, okay, so the first thing that we're going to be discussing is, so in this video we're going to be discussing main thing. What is that is the factors that are affecting your enzyme activity. Okay, and there are two factors in your syllabus. That is, number one, that is the effect of temperature on enzyme activity. Okay, and the second one is called as the effect of pH on your enzyme activity. Okay, so these are the two main factors that we're going to be studying in depth, that how, by changing these two factors, which is temperature and pH, how does it affect our enzyme activity on a specific reaction. Okay, so first of all, let's see the effect of temperature. Okay, so before I explain this, I go on to that slide, I want to come over here and I want to explain something about temperature. So you see that when I'm gonna increase my temperature, okay, so let's suppose there's a reaction taking place. Again, let's take the same example as of my previous one. A plus A is breaking down into B and C, and then there's an enzyme, let's suppose E, involved over here in it. Now, this enzyme is working perfectly fine. But what I do is that instead of keeping the temperature of this reaction constant, what I do is that I start increasing the temperature. So let's suppose that this reaction was it was proceeding at 25 degrees Celsius at room temperature. Okay. Now what I do is that I start increasing the temperature. Okay. What do I do? I start increasing the temperature. I'm going to notice two things here. Okay, the first thing that I'm going to notice is that my kinetic energy. Okay, so as the temperature is going to increase, okay, as my temperature starts increasing, what happens is that my kinetic energy. Now, what is kinetic energy, guys? Kinetic energy is how, like, it is the motion of your particles. Okay, it is literally, you can call it as how fast, not exactly how fast your particles are moving, but it is basically the energy that is associated with moving objects. Okay, so if I increase the temperature, what happens is that my kinetic energy starts to increase. Now, what of which thing the kinetic energy starts to increase? The enzyme and the substrate. Because you see, these are the two things that I have. A is my substrate, my reactant on which my enzyme is working, and the other molecule I have is an enzyme. So as I start increasing the temperature, what happens is, so this is what happens, number one. Okay, slowly as I'm increasing the temperature, my kinetic energy also starts increasing. So that means that there's some sort of a direct relationship between your temperature and your kinetic energy, which basically means that your enzymes and your substrates, they start moving faster. Okay, what do? They start moving very fast. So imagine if this is your molecule A, and then this is your enzyme. Okay, this is your A is your substrate and E is your enzyme. So if they start moving like very fastly, like this, let's suppose, like, it's going to be moving in random directions, but let's say that they start moving like very fast. Okay, so what is that going to do? That is going to be number two. Because of this, because of my increased kinetic energy, the second effect of this is going to be that increased I'm going to be having increased effective collisions. Increased effective, my frequency of effective collisions, they're going to increase. Now what do I mean by that? It literally simply means is that there are more chances of my substrate binding into my enzyme, because they're going to collide with one another very fast. Imagine like two balls, okay? This is one ball and then this is one ball. If they're going to be moving slowly, so there are going to be less chances of them colliding, right? But if they start moving like very fast, let's suppose in this direction, in every possible direction, they start moving like randomly in very fast directions, there are going to be high chances that they're going to be colliding into one another. Similarly, my substrate and my enzyme, because of high kinetic energy, why? Due to increased temperature, there are very the frequency of my effective collisions meaning that this is my enzyme, so my substrate is going to be binding into the enzyme. So this frequency of effective collisions, they start increasing. But as I said, my temperature is still increasing, okay? So this is initially when the temperature is increasing, but then comes a point when there's a max point reached, okay? So this is the, if let's suppose this is my temperature, okay, this is my temperature, and then this on y-axis is my enzyme activity, okay, how well, the rate of my enzyme activity. So you see over here that as I am increasing my temperature, okay, as I am increasing my temperature, note, okay, note as my temperature is increasing till this point, okay, only till this point, okay, my rate of enzyme activity is also increasing. Why? Because of increased kinetic energy, and then that increased kinetic energy is increasing my effective collisions. Then comes this point over here. This is called as the optimum temperature, the max rate, okay, this is literally the maximum rate of my enzyme activity at this particular temperature. So the temperature, okay, note this thing, that the temperature, okay, the temperature at which wait a second, the temperature at which my enzyme activity is the maximum, that temperature I'm going to call as the optimum temperature, okay, that temperature I'm going to be calling it as my optimum temperature. Now note that as I slowly begin to increase the temperature more, okay, so this was my optimum temperature at which my rate was maximum, okay. I think I should draw it thoda sa tamiz se because it's getting a bit messy. Wait a second, let me just remove all of this, and I'm gonna increase thoda sa, let me just write it down more neatly so you guys can really understand what I'm trying to say. So note what I'm trying to say over here, okay, this was my rate, okay, this is my rate of enzyme activity, the rate of my reaction, and then this is my temperature, okay. And the graph that I get with increasing the temperature is something like this. Oops, wait a second. This is the graph that I get, okay? Something like this. Now you need to exactly know why this graph is behaving in why this graph is behaving in such a way. Well, it is because you see, as I'm increasing the temperature from left to right, okay, this is the temperature. As I'm increasing my temperature, you guys should note that my rate is also increasing. Can you see this? As I'm increasing the temperature on the x-axis, my rate is also increasing. My y-axis is gaining height, which means my rate is increasing, but until a maximum point, okay. This is a maximum point on which my rate is actually max, and the point on which my, oh sorry, what happened, I'm really sorry, okay. Okay, so the rate, so as coming back, so the rate at which my temperature, the rate at which my the temperature at which my rate is maximum, that temperature is called as your optimum temperature. Okay, so this is called as your, this temperature right here, this is going to be called as our optimum temperature. Okay, this is my optimum temperature. Now note, okay, note one thing. As I'm moving, so as I move again, as I increase the temperature from the optimum temperature, okay, I increase even more to see if my rate increases or decreases. And you can see that the graph is telling me that now my rate is actually decreasing, my kinetic energy is increasing, there should be more effective collisions. So then why is my rate actually decreasing? That is the question to be. Well, that is because I told you guys yesterday that enzymes have a 3D shape, right? And and enzymes, they have something called as an active site. So that active site is very sensitive to temperature changes, which means that if I'm going to be increasing the temperature from the optimum temperature of the enzyme, okay, optimum temperature is the temperature at which the enzyme is working at its max rate, it's working at its best rate, okay. But if I increase the temperature from that value, let's suppose that this, I'm just giving you a random example, let's say that this enzyme works very best at let's say around 37 degrees Celsius. Now the reason why, because our body, it has a temperature of 37, so almost all your enzymes, they work best around 37 degrees Celsius, which is your optimum temperature for enzymes in your body. So if we increase the temperature from this, what happens is that the active site of your enzyme, it starts getting denatured, it starts getting denatured. What do we mean by denatured? Denatured literally means that the shape of your active site changes. Okay, so if it was initially like this, it's gonna change into, let's suppose, something like this. It literally gets um it changes its shape. So do you think now that it's going to be binding to the same substrate that it used to bind with? Like, look at this. This was a substrate that it was initially binding to, because the substrate and the active site, they both were complementary to one another. But now look over here. The substrate can no longer bind to my active site because my active site has denatured. Why? Due to such extreme high temperatures. Okay, so the active site actually loses its specific shape. But remember, the kinetic energy is still high. Why? Because temperature is directly proportional. Remember, temperature is directly proportional to kinetic energy, so my kinetic energy is still very high. But it is just that my effective collisions have decreased in this point. Why have my effective collisions decreased? Because the substrate can no longer bind properly to the active site of my enzyme. Okay, so the fit between the enzyme and the substrate, it has greatly reduced. Okay, so that is what the whole scenario is regarding temperature. Okay, I hope you guys have understood it. So again, I've also added this diagram for you guys to understand that. As you can see, this is how a normal enzyme work. Okay, this was your enzyme and this is your substrate at normal temperatures, at the optimum temperature, what we call it, or even low, even at lower temperature that is lower than the optimum, the enzyme still works. But it's just that because the effective collisions are so less that they're not able to bind to one another properly, तो उसकी वजह से भी your rate decreases. But as the temperature reaches to your optimum, your enzyme rate is the max. Okay, it works the best. But as you increase the temperature beyond your optimum temperature, what happens is, you can see over here, the active site has denatured, it has changed its shape, it has lost its shape. So now my substrate can no longer bind to my enzyme. Okay, Similarly at low temperature, can you see at low temperature my enzyme shape is unchanged? Can you see why? Because enzymes only denature, the shape of my active site denatures upon high temperatures, but in low temperatures, even though my active site is intact, okay, there is no there is no change of shape in my active site. Can you see this was my active site before, and then by exposing it to a cold temperature, the active site still remains the same. But what happens over here? The effective collisions reduce. Why? Because the kinetic energy of my enzyme and my substrate reduced. So if they're not going to strike with each other at such high speeds, there is very less, there's a very less chance of them actually colliding into one another and binding into one another, which means that my substrate and which means that my enzyme substrate complex is not going to be formed. Hence, the substrate binds very less often. So your enzyme is actually inactive. Okay, so your enzyme is less active. So that is also the graph that I told you guys. That is the same graph. If you guys want to, make notes from here, so you can definitely use this. This is all that you need to know about the effect of temperature on the rate of enzyme activity. And just note that the optimum temperature of enzymes in our body is 37 degrees Celsius, because that is our body temperature. Why our body temperature is 37 degrees Celsius? Because this is the optimum. This is the temperature at which the rate of my enzyme activity is max. Okay. Now, moving ahead towards the second part, which is the effect of pH on the enzyme activity. This effect of pH on the enzyme activity is exactly similar. Okay, it is exactly similar to how we did the effect of temperature. Okay, what you need to know is just like how every other enzyme has an optimum temperature, similarly, every enzyme works on an optimum pH as well. Okay, there's a certain pH that it works. Now, not every enzyme works at the same pH. For example, the optimum pH for the enzymes in your stomach, they will be acidic, right? Similarly, you don't need to remember this, but just telling you that um, your pH for your optimum pH for every enzyme in your body is different. Okay, so the idea behind is that at optimum pH, what is an optimum pH? An optimum, let's say that this enzyme works at an optimum pH of around 6. Okay, so that means that at 6 it has the highest chance of collision between the enzyme and the substrate. Effective collisions increases, and plus there the as you can see, the active side of my enzyme, it does not denature, because again this is the optimum pH. But as I increase the pH from 6, or if I decrease the pH from 6, okay, if I increase the pH from 6, let's suppose I increase it to 11, or if I decrease to a pH of, let's suppose, 3. Okay, this is just an example. But yeah, if I increase or decrease the pH from the optimal pH, what happens is, look at over here. This is how my enzyme was before, uh you know before before I changed the pH. And as soon I made the pH high, as soon as I made the pH alkaline, what happened? The active site, it got denatured. Yes, my active site got denatured. Again, if it got denatured, then what will happen? My substrate is no longer going to be binding to my enzyme. Okay, and similarly, if I made the pH too acidic, if my pH is going to be too low, it means that it is acidic. So then what happens? Again, my active site becomes denatured, and if it becomes denatured, then my substrate will no longer bind to my enzyme. Okay, and so the reaction is going to be, the rate of reaction decreases significantly. So again, this is also the same pH. Okay, the optimum pH for most of our enzymes is 7, but again it can vary for every enzyme. So if it's going to be too alkaline, my enzyme, again, the enzyme activity decreases. Why? Because the active site denatures. And similarly, if the pH is too low, if it's too acidic, then also my enzyme activity, my rate of enzyme activity, it also decreases. And you see, it starts to decrease. So that is all for this lecture. And that literally wraps up our entire chapter. So I hope you guys enjoyed this chapter, and I'll see you in another interesting video, in another interesting lecture. So see you then.